What's going on, folks? Justin Schaefer here, also known as Mr. Fascinate. I have the ultimate pleasure of joining one of Regeneron's own today in the fight against COVID-19. Alina Baum is a senior staff scientist at Regeneron Pharmaceuticals. She is the lead scientist on the COVID-19 antibody program that is on track to enter clinical trials in June 2020. She received her PhD in molecular virology from the lab of Adolfo Garcia Sastre at Mount Sinai School of Medicine and performed postdoctoral studies with Charlie Rice at Rockefeller University. As a fellow nerd, I'm so amazed with the work that you guys are doing and obviously the importance of science in the context of all of this. And one of the things that I want to preface our interview with is that this was initially a partnership with Regeneron around the Science Talent Search, which is going to be an in-person event that we did, but I had to pivot to highlighting the company's work against COVID-19 because of the situation that we've been placed in. Alina, first things first, what is a day like in the lab for you?
Well, so yeah, it's been a huge, I think like every person on the face of the planet, we have been really impacted by the pandemic. So I haven't actually been on campus since mid-March. I've been working from home. Luckily, I can do most of my work from home because I work on the computer most of the time. But my team, is still working in the lab. So the challenging thing for us has been that obviously when you work as a team, typically we see each other, we talk to each other on a daily basis, we check in quickly. We haven't been able to work like that. So we have to find a new way to communicate. For myself, I think the most challenging thing has been working from home with my kids. A lot of parents can relate to this. I have two small kids, boys that have been out of school and out of daycare. So we've had to find a way to make that work as well.
Yeah, I can imagine what you're going through. I mean, having to focus on things that are, so detail oriented, and then, you have a kid here that
there's a lot of bribing going on and a lot of iPad time.
Well, I guess you got to do what you got to do, right? So who are some of the people that are on your team? What are their responsibilities and what do they do right now?
So I have a fairly large team. There are about 10 people in my group. A lot of them have been working on the COVID work. They are, all of them really, they work in the lab, so they go into the lab, they run the experiments, they generate the foundation of the data and that really leads to us to be able to develop these drugs. So these people, they are still going in the lab on a daily basis because even though we have identified what our clinical molecules are going to be, we're producing them now for, beginning of our clinical trials, we're not done with research. So there's still research is ongoing, full force. We're trying to understand the biology better. And they're going in and running experiments, day to day, even under these really tough circumstances. And they were doing this back in end of March, beginning of April. We're in New York, obviously. The pandemic was in full swing here. So these guys deserve a ton of credit because none of this would have happened without their work.
Oh, that's incredible. That's amazing. And I know a lot of young people that aspire to be scientists are following me as well as educators. So, it's an important testament to, if that's the life that you choose, those are the kind of sacrifices that you have to be willing to make for situations like this one. And so, what I'm curious about is, it's really interesting to me, for a virus that's so infectious, for people to actually be handling it on a daily basis and obviously minimize risk and minimize exposure. So what kind of safety precautions do those scientists or the folks that are working in the laboratory have to undertake?
Yeah, so we at Regeneron, we're not actually working with the SARS-CoV-2 virus itself. We work with collaborators that work with that virus when we need to generate data with the virus, because working in that virus requires what's called a BSL-3, Biosafety Level 3 lab. Don't know if you're familiar with this, but there are 4 levels of biosafety labs, from 1, which is like nothing, all the way to 4, and that's like Ebola. So SARS-CoV-2 is not quite as bad as Ebola, but it's a BSL-3 pathogen, so it does require extra precautions when you work in a BSL-3 lab, such as, for example, you always have to work with somebody else. You're not allowed to be in the lab by yourself. You minimize the use of syringes, things like that. Some BSL-3 labs, you actually have to shower to go into the lab and to go out of the lab, and you have to change your clothes. So But what we have been able to do, because Regeneron has an infectious disease group, and we've been working on infectious diseases for a long time. So we have people who have a lot of expertise with working with other viruses. We've been able to make kind of surrogate viruses that mimic the SARS-CoV-2 virus, but allow you to work in a BSL-2 setting, which is what we do. So we've been able to generate a lot of the critical data that we need for this program ourselves. But we do collaborate when we absolutely have to with other labs.
Right. That's really, really cool. So a surrogate or like a pseudo virus. So
Yeah, exactly.
what exactly is that? Like from a molecular perspective, how's that different from a virus?
It's really interesting. So what we've done is there's this virus out there called vesicular stomatitis virus. And this is kind of this, virus nobody has ever heard of because it's really a virus of cows and some other livestock. But it's a very, very common lab virus. People have been studying it since like the 50s, so we know how to work with it really well. And what we can do is we can play around with the genome of this virus. We can take out the protein on this virus that mediates attachment to cells that it infects, and we just replace it with a spike protein from the SARS-CoV-2 virus. So now you have a virus that can replicate, that can enter cells. It uses the exact same way to get into cells as the actual SARS-CoV-2 virus, but it's now safe. So that's how those systems work, and they really allow us to test the potency of the drugs that we're developing because we're targeting the same exact molecule that you would target on the actual virus.
That's really, really interesting. So essentially the contents of the virus itself are not as damaging, but the methods that the virus uses to get into cells are exactly the same.
Yeah, it's a chimeric virus, right? You bring two different really pieces together, the guts of the virus, which are coming from VSV, and then the outer surface is coated with what, the SARS-CoV-2 glycoprotein spike.
Really, really interesting. So Regeneron has this approach, right, to developing these preventative antibodies for COVID-19. So can you outline what that approach is?
Yeah, so Regeneron really is an antibody-based company, although we do work on a lot of other things, but our bread and butter are antibodies for a lot of different diseases. The thing about antibodies and the way they work, most of them, the virus, in order to enter a cell, attaches with its surface protein to a receptor, which is on the surface of the cell that it's going to infect. And this interaction is absolutely required. So you need for these two things to come together. The virus can get internalized, it starts to replicate, makes, millions more copies of itself. And that's how it makes you sick, it spreads. What antibodies do, the very best, meaning most potent antibodies against viruses, is they block that interaction. So now that spike protein, which is on this virus, and then the receptor, which is called ACE2, they cannot come together anymore. And what the beautiful thing about antibodies is they can work both to prevent infection of someone who is not yet infected, but they can also treat someone who is already infected by blocking the spread of the virus in that infected person. They do more things, and it's never this simple, but this is kind of like the really basic way antibodies work.
Right, the really high level. So there are multiple mice, right, that are being tested and used, and we're trying to figure out which antibodies are the best. And so the best ones, they'll block the proteins. So can you give me
the way we figure out which antibody, right? So what we did this time is, you mentioned mice. So Regeneron, a long time ago, way before I was there, they developed what are called humanized mice. These are mice that make fully human antibodies. So they've been genetically engineered to make an antibody that has everything coming from a human. And this is important because these antibodies can be given to people and they're not immunogenic. And they work the way that antibodies are supposed to work in native human antibodies. So what we've done for COVID-19 is we, as soon, and I have the date in my head because it was like January 13th, I think, we got the sequence of the virus. And as soon as we got the sequence of the virus, it was sprang into action. We generated immunogens that then went into these mice, and they basically primed the immune response of these mice to make antibodies, like a vaccine would. We basically vaccinate the mice. And then we have this platform, that has been developed over decades in Regeneron to screen thousands and thousands of B cells for different types of antibodies, and then we put in what we call screening functional assays. So we test thousands of these antibodies for function, and the most critical function that we were screening for this program is the ability to neutralize the virus. All of that is done in vitro. So we're not doing this in mice, we're doing everything in cells and looking to see if we can block the entry of the virus into the cells. Once we pick out the most potent antibodies, we call these our leads, then we start, doing even more experiments. We're doing some animal model experiments, but basically, really, it's all based on this neutralization potency is how we were able to identify our antibodies, and also looking for antibodies that can be combined together to make a cocktail, which is another thing that's really important, because we're trying to get antibodies that can bind to the virus at the same time in two different places. And that is important to safeguard against potential escape that you worry about with RNA viruses.
Right. So then this would be used as a vaccine, or is this something completely different from a vaccine?
Well, so you can use them in different ways, right? So it kind of works as a vaccine because you can prophylax people, right? You can dose someone who's not infected in order to prevent infection. The difference between giving someone antibody and giving someone as a vaccine is with an antibody, you don't rely on that person's immune system to respond. You're basically giving them the thing that's going to protect them from getting infected. And the antibodies will work almost immediately as soon as you dose someone. For a vaccine, what happens is, you have to give someone a vaccine, but then you wait for the immune system to respond to the vaccine. And that takes usually at least, a couple of weeks, usually 3 weeks. Sometimes it requires multiple doses. It doesn't work in everybody. So some people, if they're immunocompromised, or especially the elderly, sometimes don't respond very well to vaccines. So the mechanism of action can be quite similar, but they can be used in different settings and they can complement each other.
Interesting. That's interesting. Can you highlight that relationship a little bit more, Alina, about how, what's the best use case for a vaccine versus this antibody treatment, or both?
Well, another thing that's important that I didn't mention is, for an antibody treatment, that's not a permanent treatment, right? So for an antibody, you will dose someone, if you have a really potent antibody, you could potentially protect them maybe even for months with a single dose. But that's really your best case scenario. You're very unlikely to protect someone for a year with a single dose of antibody, because those antibodies will get cleared of your body eventually, and you're only protected as long as you have enough antibody on board. With vaccines, what you're trying to do is establish really a long-term immune response. Some vaccines give you almost lifelong immunity. And you can vaccinate yellow fever, for example, is a very good vaccine. Measles vaccine, although not lifelong, but you can protect someone for a decade. Other vaccines, not so much. You have to revaccinate someone, every year, for example, for influenza. And, the funny thing is we don't necessarily understand why. So that's another major difference. If you want to, establish something that mimics herd immunity, everyone has heard about herd immunity, and that's the thing that you need to have in order really to stop the spread of the virus. You're trying to do that with a vaccine by covering a huge percentage of the population of the world. That's where a vaccine will become really important. With an antibody prophylaxis, what we hope for is, one, that it can be a stopgap before vaccines are available, right? We're waiting for these vaccines. We still don't have efficacy data. We're all hoping that maybe later in the summer, or maybe in the fall, we start getting some glimpses whether some of these vaccines that are being tested in the clinic now work, but they're not yet out there. So there's this possibility of using antibodies that are, that will be entering into clinical trials in very soon, and using those until there's widespread use of these vaccines.
Really, really interesting. That's really interesting. So, and this is kind of more of, I guess, my geeky theoretical side coming out with this question. So could you guys develop preventative antibodies that could self-replicate within the human body to potentially last longer? Or is that something
that's an interesting idea, and kind of. So what people have been working on is, these other platforms to make antibodies. So some people are trying to develop almost like gene therapy platforms. So there are these viral vector platforms, for example, adeno-associated virus, AAV is one of them, where you deliver a DNA. So instead of delivering protein, which is your normal antibody drug, you deliver a DNA. And the DNA now encodes for this antibody. So what you're hoping to do is by delivering this DNA, your body starts making this antibody all by itself. And because the DNA can stay in your body for a very, very long time, maybe years, that it's a continual source. So you're almost turning your body into like this production factory without requiring a vaccination.
Man, that is fascinating. You are painting some vivid images in my mind. So hopefully, some
Things
vivid
are
images.
happening. That's the
Yeah,
amazing thing, you know.
yeah,
This is real.
yeah. It almost sounds a little bit like science fiction, but
Well,
it's
you know what's going to
amazing.
be one of the things that will be really interesting, I think, from this pandemic is that there's a lot of really new, exciting technology that hasn't really been tested in the clinic yet that we're going to get a chance to see how they're working. So some of these are these RNA vaccines that maybe you've heard about, and maybe some of these AAV type platforms to make vaccines. So I think, you know, a year, 2 years from now, we're going to be way further ahead in understanding which of these platforms are really promising and maybe which ones are not.
Hmm. Yeah, that sounds super fascinating. I mean, that's one of the things that you don't think of a lot of times is in a situation like this, innovation is necessary, and you know it could have lasting effects, lasting positive effects for humanity in terms of our fight against viruses for the
Oh,
future. So,
absolutely.
yeah, very, very interesting stuff. So, all right, so let's fast forward. Let's say, you know, 3 months after an antibody approach has been fully developed and tested, what do those first 3 months look like?
So I think, you know, I've never wished for a crystal ball more than I do now. I think the really, really honest answer is I have no idea. You know, nobody has an idea. We don't know what, you know, October is going to look like, right? But what I hope is that there's a drug out there that people know works, that takes some of the fear out of this virus. And by running, you know, a controlled clinical trial and having really solid data, people can know that, you know, if they do happen to get infected, that there's a treatment out there. And also, you know, we hope that with prophylactic use, what we can do is we can offer some immediate protection to some of the most vulnerable people, right? Whether it's healthcare workers, first responders, essential workers who can't socially distance, elderly, obviously, you know, people in nursing homes, people that are immunocompromised, you know, people that are really scared now to even go outside. You want to provide some kind of a relief, which I think will have a huge benefit on you know, the world really. So that's the hope. That's what we're working towards.
Well, I can certainly appreciate your optimism, Alina, in these weird times. I think that people like you are absolutely the heroes and the sheroes, and I'm just so glad. Yeah, I'm so
You know,
glad.
we're all, you know, what I think we're all doing what we can. You know, that's been an amazing thing. I think we all, whatever background we have, whatever we can do, we're just trying to do something to make things better. And everybody's effort is important. You know, what we're doing is important. What vaccine companies are doing is important. All the people that are really studying basic biology, trying to understand the disease, that's invaluable. And the academic community has, you know, given us so much in like the last 6 months, because I think we all understand that the way for us to get back to anything that looks like normal is going to be through science. You know, there is no alternative way. The alternative way is to wait until 70 or 80% of the population has been infected, and we know what kind of a toll that's going to take. I don't think we want that to be the way out. So everyone's working, you know, and doing what they can.
Right, right. And as someone that's been an advocate for science for most of my adulthood, you know, I think that, you know, this is something that young people, educators, people all in all walks of our community and my following especially need to hear. So thank you, Alina, for those optimistic, authoritative words on COVID-19. Alina, was there anything maybe you wanted me to touch on?
Well, you know, I think there are some things that I think are important to keep highlighting, even though they've been many highlighted by others before. You know, there are a lot of misconceptions, a lot of false information that's out there on the internet. I think it's very important to keep speaking the truth, keep focusing on things that are important, you know, not supporting some of these conspiracy theories.
All right, 100% agree with you on that one. So anything else you'd like to share with our audience, maybe some parting words, you know, some words of encouragement?
Parting words, I would say, you know, for all of those young people that are trying to figure out what they want to do with their lives and are kind of thinking about science. It's a funny thing, when I was, you know, I was taking some classes when I was an undergraduate, and, you know, one of my TAs, I think, for a biology class asked me, oh, have you considered majoring in biology? Because I liked it. And I was like, well, you know, everything in biology has been done already. There's nothing new. It was like the most ridiculous, naive thought, where I really didn't maybe understand the scope of the unknown. So what I would say to people that are thinking and are kind of interested in science is that there's so many things that you can do with science. And if you have a passion for it, and if you think you're good at it, you should jump in, because it's an amazing thing to do. It's an amazing field to be in. You know, I'm like so fortunate to be able to do what I do and to be excited about my work on a daily basis. I think it's a really incredible field, and hopefully there are many, many opportunities for young people that want to get into it.
Oh my gosh. Yeah. I mean, yeah. And I can tell, Alina, even, you know, on your end, I can see the curiosity, maybe that's been with you since you were a kid, is still deeply within you, and it kind of propels you.
Oh yeah. It honestly, it just grows, because we don't know anything. All the questions are out there. So yeah.
Yeah, and that's why I have so much respect for the scientist's approach. It's like the acknowledgement of, look, here's what I've studied, here's what I experimented on, and here's what we don't know yet. So
Yes,
they're always advancing that frontier. Well, thanks again, Alina, for sharing these wise words, sharing that message with our audience, and I super appreciate your time.
Thank you, Justin. This was really fun. Thank you.